So we are going to be completing Chapter 9. This is the final part, Part C, for muscles and muscle tissue. If you have not viewed Part A and Part B, please be sure to view those as well. You can find it in the links in the comments below. In addition to that, they are on the playlist. Okay, so the force of muscle contractions, it depends on the number of cross bridges that are attached, and this is affected by four factors. The very first one is the number of muscle fibers that are stimulated or recruited. So the more motor units you have recruited, the greater the force is going to be. Number two is the relative size of the fibers. So the bulkier the muscle, the more tension can develop. So muscle cells, they can increase in size or hypertrophy with regular exercise. So when you're going to the gym and you're lifting weights, you're increasing the resistance. What you're doing is you're causing these muscle cells to grow, to increase in size. And again, this is what we refer to as hypertrophy. So number three is the frequency of stimulation. So when a muscle is stimulated rapidly, the contractions are summed and they end up becoming stronger and more vigorous. Ultimately, they end up producing tetanus. Now, to understand number four, we have to think back to the very first lecture on Chapter 9. And when we were discussing about the A bands and the I bands and the thin and the thick filaments. So within the sarcomere, the ideal length-tension relationship, this occurs when the muscle is slightly stretched and the thin and the thick filaments to overlap optimally. Because this relationship, it permits sliding along the entire length of the thin filament. Now, if the muscle fiber is stretched so much that the filaments do not overlap, then the myosin heads have nothing to attach and they can't generate tension. On the other hand, if the sarcomeres are so compressed and cramped that the Z discs, they abut the thick filament and the thin filaments, they touch and interfere with one another, you're going to end up with either very little or no shortening at all. And with this, we see identical relationships that exist in whole muscles. So when we have muscle fibers with sarcomeres that are between 80 to 120% their normal resting length, these generate more force. So if we have sarcomere that's less than 80% of their resting length, then the filaments overlap too much and the force decreases. On the other hand, if the sarcomere is more than 120% of the resting length, then the filaments do not overlap enough, so the force decreases. Now, this graphically represents the factors that increase the force of skeletal muscle contraction. So again, you see that when we have a large number of muscle fibers recruited, the contractile force will increase. When the muscle fibers are large, the contractile force will increase. When you have a higher frequency of stimulation, again, you're going to have stronger contractile force. Then also, when you had the muscle and the sarcomere stretched, they're to slightly over 100% of their resting length, then you also end up getting this stronger contractile force. And what this is showing you is this in this picture. So when you have what did you remember what we said? The optimal sarcomere operating length is between 80 to 120%. So when it's between 80 to 120%, what they're trying to tell you is this. These myosin heads over here and the actin, they're able to attach and pull themselves entirely, okay? So now they can take advantage and forcefully contract because they have all this area to play with. However, when they're pulled too far apart, okay, as they're showing you over here, then they're too far out to stretch. So this entire thick filament is not going to be sliding over here. It's not going to be within optimal working conditions. In this over here, what you have too much overlap, okay, when it's too short, when it's less than 75% and less. And what's going to end up happening is, what are they going to bind to over here? They're already at the Z disc. It's already at the Z disc. So you're not going to get much, you're not going to end up getting good contractile force because this is too short. This is too long. So you want to stay right in between. Muscles vary in how fast they can contract and how long they can continue to contract before they fatigue. These characteristics, they're influenced by muscle fiber type, load, and recruitment. Now we're going to be using two major functional characteristics to identify muscle fiber types. Now the first one that we're going to be looking at is the speed of contraction, which are the slow fibers and fast fibers. Now the differences in their speed, it reflects how fast their myosin ATPases split ATP and on the pattern of electrical activity of their motor neurons. The other characteristic is the pathway that they use for forming ATP. Now, the cells that rely mostly on oxygen using aerobic pathways for ATP generation, these are the oxidative fibers. And then when you have the fibers that rely more on the anaerobic glycolysis, these are the glycolytic fibers. Based on these two criteria, skeletal muscle fibers, they can be classified into three types: slow oxidative fibers, fast oxidative fibers, or fast glycolytic fibers. Most muscles, they contain mixtures of fiber types, which ends up resulting in a range of contractile speed and fatigue resistance. Now, all fibers in one motor unit, they are the same type. It's the genetics that dictates an individual's percentage of each type of these fibers. Now, different muscle types are better suited for different type of jobs. Now, slow oxidative fibers, they're best for low intensity endurance activities. So like maintaining posture or for going, for a marathon, for example. Fast oxidative fibers, they're good for medium intensity activities like sprinting or walking. Now, fast glycolytic fibers, these are best for short-term intense or powerful movements. So, you're taking a ball and you're hitting a baseball, or you're taking a ball and you're a quarterback, you're playing football and you're throwing, making a 90-yard pass. So this is what these fast glycolytic fibers are good for. This image is showing you factors influencing velocity and duration of skeletal muscle contraction. So if you have a predominance of fast glycolytic fibers, your contraction velocity is going to be high. Also, if the load is small, the contraction velocity is going to be high. If there is a predominance of slow oxidative fibers or these fatigue resistant fibers, the contractile duration is going to be long. Okay, it's going to be high. So when you look at this diagram over here, this table, what it's trying to show you is, first of all, the three different types of fibers, and then it's showing its metabolic characteristics. Instead of memorizing this, try to understand why these properties are that the way they are, and why you end up getting its best use for. So for example, in slow oxidative fibers, the speed of contraction is slow because the myosin ATPase activity is slow, right? The primary pathway for ATP synthesis is going to be aerobic, right? Slow oxidative. So remember, why is this going to be slow oxidative? Because first of all, it's using oxygen, so it's aerobic. And remember, when you have aerobic, you're going to end up having a lot of mitochondria. And remember, when you end up producing ATP through glycolysis and the Krebs cycle and electron transport, it takes a long time. So again, it's another reason why it's going to be slow. But remember, you end up getting a lot of ATP that ends up yielding. So right over here again, so the primary pathway for ATP synthesis is aerobic. Now remember, when you have aerobic, you need oxygen, right? So when you need, if you're going to be using a lot of oxygen, you need to have a lot of oxygen stores, and this is where the myoglobin comes in. So remember, if you guys remember, myoglobin is it's a storage protein for oxygen. So which is why you also have why it appears red in color, which is also another reason why you need a lot more blood vessels. Okay, so blood carries oxygen and carbon dioxide to and away from the cells, in addition to that, nutrients. So you're going to have a lot of capillaries there as well in slow oxidative fibers. Also, you're going to have it's going to be its recruitment order is going to be the first one, and its rate of fatigue is going to be slow. Now, glycogen stores, it's not going to have too many glycogen stores. Why? Because this process is occurring slowly, so it's not it's not very it's not a big gas hog. Okay, so it's think of this as kind of being like a Toyota Prius, right? It can go for a long time. It's very fuel efficient, so it doesn't need too much fuel. It doesn't need too many fuel reserves also. So on a small tank of gas, all right, it can go a very long way. And so what is this good for? Well, this is good for endurance type of activities like running in a marathon, okay, maintaining posture. The fiber diameters, they are small here, and we already covered these details. So now let's go to the second time. It's fast oxidative fibers. Now, fast oxidative fibers, this is also an aerobic pathway, which means it requires oxygen. So you're going to see, because of that, we're going to see a high myoglobin content. So because we have a high myoglobin content, we're going to have many capillaries as well, and then the color will also be a red to pinkish in here too. Now, what else? Intermediate amount of glycogen stores. So this does use more energy than this type of than the slow oxidative fiber does. Uh, but yeah, the speed of contraction is going to be fast because the myosin ATPase activity is fast, right? Remember what I said, these two things, they go hand in hand. The myosin ATP activity, it affects the speed of contraction. It's one of the things that affects the speed of contraction. Um, and we told you that this is aerobic. It uses the aerobic pathway for generating ATP, although it does use some anaerobic glycolysis too. Its recruitment order is going to be second, and its rate of fatigue is intermediate. So again, it's moderately fatigue resistant. The fiber diameters, they are large, and the mitochondria, again, you're going to have a lot of mitochondria here. You're going to have a lot of blood capillaries here as well. Um, yeah, notice that there's an asterisk over here. So again, we said is the fiber diameter was large. In humans, they're not the case, but in other animals, we found them. They're found to be much larger. But in us, not the case. Now let's go to the last one. This fast glycolytic fibers. Okay, so remember, fast oxidative, slow oxidative. These both require oxygen. Okay, think of it as oxy. Think of it being meaning oxygen, so they're aerobic. Now, fast glycolytic, there's no oxy in this word, so that means they don't need oxygen, so it's anaerobic. Now, its speed of contraction is fast, and it's fast because the myosin ATPase activity is fast also. It's going to have a low myoglobin content because why? It doesn't use oxygen. Okay, it's not required, so that's why it has a low myoglobin content. The glycogen stores now, it's going to be high. So this is a gas hog. Think of it like that. Its recruitment order is a third. Its rate of fatigue, it's fast. Okay. Mitochondria are few. So doesn't have. Will it have a lot of mitochondria when because what happens? Remember what we said. When you have through the ATP through the energy production process. This will only go through glycolysis and then it stops again. That's why it says anaerobic glycolysis. It stops over here. So the pyruvic acid is not able to enter the mitochondria. So therefore, you're not going to have too much mitochondria over here. Whereas over here and here, you will. Myoglobin content is going to be low also. And the reason that myoglobin content is low is because, again, it's anaerobic. It's an anaerobic process. Not too much oxygen is going to be used here. So why is it going to store the myoglobin? Glycogen is high. Okay, it needs a lot of glycogen. It needs a lot of glucose because this is a gas hog. It's just going to keep using. Remember, through this process, glycolysis, it only nets 2 ATP, right? So very little energy is produced. So therefore, it needs to have a lot of fuel. With this one, think of it this way. If you want to imagine one glucose molecule kind of equating to like a gallon of gas, all right? So if over here, if 1 gallon of gas is going to take you 32 miles, think of it like that. In this case, 1 gallon is only going to give you 2 miles. Okay, so what's going to happen? It needs a huge tank. Okay, so you need to have a lot of glycogen stores. And again, people don't really want to drive this gas hog, so it's going to be the third. Recruitment order is third, but its rate of fatigue is fast also. Okay, it's highly fatigable. What is this good for? Short-term, intense, or powerful movements. So, as we said earlier, hitting a baseball, taking a ball and throwing it like a football and throwing it across the field for a touchdown. Its fiber diameter is intermediate. Again, it has few mitochondria, as we described earlier. Also, you're going to have few blood capillaries coming there. Its color is going to be pale as well. Why? Because, again, low oxygen. So when we look at load and recruitment, this is quite simple. So when it comes to load, muscles, they contract fastest when no load is added. So the greater the load, the shorter the duration of the contraction. And the greater the load, the slower the contraction takes place. Now for recruitment, the more motor units are contracting, the faster and more prolonged the contraction is going to be. Okay, this is just common sense. Now these graphs, they show you the influence of load on duration and velocity of muscle shortening. So this very first graph, it's showing you that as the lighter the load is, okay, which is showing you by green, and this is the heavier load, which is by blue, and then you have an intermediate, which is the orange. So the lighter the load, the distance the muscle shortens much more, as well as the duration in time, it's going to be much longer. And as the load increases, the distance is much shorter as well as the time. And this graph shows you the velocity of shortening decreases as the load increases. Now, in aerobic exercise or endurance exercise like jogging, swimming, or biking, uh, these type of exercise, they lead to increased muscle capillaries, increase in number of mitochondria, as well as myoglobin synthesis. Now, you have to understand why this is the case. In these athletes, now remember, these are trained professionals. We're not just talking about, the average person in this classroom who decides to start, jogging tomorrow or for the next few days. Not going to happen. But if you keep up at it, over the course of several weeks to months to years and beyond, you're going to start to see these things happen. And then they're going to be sustained results. Meaning, you have to keep up with it. Otherwise, if you don't use it, you lose it. But what happens is this. Your muscles will start making more capillaries. And the reason your muscles making more capillaries is for two main reasons. Remember, you want to get a lot more glucose getting to the muscle cells. In addition to that, you want to have a lot more oxygen and carbon dioxide being exchanged as well. So for the oxygen part, myoglobin is the storage protein for oxygen in muscle. So this is why your body's going to start, these muscles, they're going to start producing more myoglobin in order to store this extra oxygen there. In addition to that, there's going to be an increased number of mitochondria. Now remember, this is an aerobic pathway. So the ATP that's being produced is being produced through the aerobic pathway, meaning that when the glucose is broken down, it gets produced into that pyruvic acid. Then the pyruvic acid enters the mitochondria. So remember, this pathway aerobically, you are going to be netting up to 32 ATP molecules. And in order for this to happen, you have that pyruvic acid has to enter the mitochondria. So again, in these individuals, their body, it produces lots of mitochondria to be able to produce this high amount of ATP. Now, this results in greater endurance, strength, and resistance to fatigue. So again, this is why somebody can jog for 3 miles, 4 miles, with quite relatively ease and not fatiguing too much. Again, comes after a long period of time. This is after training for months. It's not going to happen overnight. It's not going to happen within a week, but it's going to take several weeks, to achieve these results. Also, what could happen is, again, if you're training, some of these muscle fibers, they could convert from fast glycolytic fibers into fast oxidative fibers. Again, because what's going on? If you're starting to use more, if you're doing much more endurance endurance exercises, then your body is going to need more oxidative fibers. It's not going to need these fast glycolytic fibers, because again, what you're doing is endurance, so it needs fibers that work well with this type of activity. So your body's going to produce what it needs, not what it does not need. Now, when you do resistance exercise, now these are typically anaerobic. So when we're talking about weightlifting, isometric exercise, now these lead to muscles getting big, okay? So we call this muscle hypertrophy. And again, this is due primarily to the increase in the size of muscle fibers. Now, a lot of people think, oh, I'm getting more muscle, I'm getting more muscle. No, you're not getting more muscle fibers, okay? The muscle fibers stay the same. Your muscles are getting big, okay? They're just getting bigger. They're hypertrophying the muscle cells. So in addition to that, you're going to see an increase in mitochondria, increase in myofilaments, glycogen reserves also go up, glycogen stores, and as well as connective tissue. So what this ends up resulting is an increase in muscle strength as well as muscle size. Now let's look at some homeostatic imbalances. Now muscles, they have to stay active in order for them to stay healthy. Disuse atrophy, also known as degeneration and loss of mass, this could be to immobilization or loss of neural stimulation. And this can start almost immediately. For example, if you end up fracturing your arm. Now, when you break your arm, you end up getting your arm put into a cast, and that cast will then, you put your arm into a sling, and now you're not moving for the next 4 to 6 weeks. Then what ends up happening is muscle strength, it starts to decline at a rate of 5% a day. Paralysis also can end up resulting in this also. So when we're saying a loss of neural stimulation, this is what we're talking about. So these paralyzed muscles, they may start to atrophy, and then they end up shrinking to about 1/4 their initial size. Now, in the case of when you have a fracture, after the bone is healed, then you can go back to physical therapy, and then they can restore normal function of that limb, of those muscles. But again, when you have some paralysis going on, what could end up happening is fibrous connective tissue can end up replacing that lost muscle tissue, and at that point, rehab is impossible at this point. Now, something else that I mentioned, which does not relate to this, but again, it's kind of goes along with what I said. That if you don't use it, you lose it. So when you look at these individuals that are building muscles, that are bodybuilding, um, if they've worked out for the last 9 months and then all of a sudden, the holidays are coming and they start eating bad and, they go visit their family and, for the next 2 to 3 weeks or 4 weeks, they're eating bad. In addition to that, they're not exercising at all. When you see these individuals, when they return back at the gym, you'll notice that they're not as big as they used to be. In other words, their muscles have already shrunk. So muscles, you lose them really quickly. It takes them a long time to build up, but you can lose them very fast. And that's it for skeletal muscle. Now we move on to smooth muscle. Now, smooth muscle, we find it in the walls of most of our hollow organs. The exception being is the heart. So when you look at your stomach, your large intestines, your small intestines, your esophagus, your trachea, blood vessels, all these organs, they're all lined with smooth muscle. Now, the exception is the heart. The heart, it has its own muscle type, which is cardiac muscle. Now we're going to be looking at the microscopic structure of smooth muscle fibers. So smooth muscle fibers are spindle-shaped cells of variable sizes. Typically, they're going to be thin and short compared to that of the skeletal muscle fibers. They have a centrally located nucleus, and unlike skeletal muscle and cardiac muscle, they lack striations. Smooth muscles, they lack the coarse connective tissue sheets that we've seen in skeletal muscles. However, they do have an endomysium. Now, all but the smallest blood vessels, they contain smooth muscles that are organized into two layers of opposing sheets of fibers. There's a longitudinal layers where the fibers, they run parallel to the long axis of the organ, and when this layer contracts, it causes the organ to shorten. Then there's a circular layer, and in the circular layer, the fibers, they run around the circumference of the organ, and when this layer contracts, it causes the lumen of the organ to constrict. So with both of these alternating, contracting and relaxing, this is what forms this peristalsis to occur. And peristalsis, again, this is the alternating contractions and relaxations of the layers that mixes and squeezes the substance through the lumens of the hollow organ. And in this illustration, we can see this is a cross-section of the small intestines, and what they're showing you here is this is the longitudinal layer of smooth muscle, and this is the circular layer of smooth muscle. So this is the intestines. So the intestine starts right over here. It's connected to the stomach. This part's it's called the duodenum. And what happens is this: when you put food into your mouth, it's no more, and you start chewing it, it's no longer called food. It's called the bolus. So as the bolus moves down this esophagus, enters the stomach, it's now called chyme. So when the chyme enters the duodenum, the first part of your small intestines, what's happening is this in this illustration. Actually, remember, all this is lined with smooth muscle. So even starting from up here, and then throughout the stomach and inside here, these alternating contraction and relaxation of these muscles, it pushes the food forward. Okay, it's pushing it forward. When one part contracts, it moves forward, the other part relaxes, so then it can enter. One part contracts, other part relaxes. So this way, it just kind of pushes the food, it moves along a way in a somewhat controlled manner. Smooth muscles, they don't contain any sarcomeres, myofibrils, or transverse tubules. The sarcoplasmic reticulum of smooth muscle fibers is much less developed than that of skeletal muscle fibers. In addition to that, they lack a specific pattern relative to the myofilaments. Now, some sarcoplasmic reticulum tubules of the smooth muscles, they end up touching the sarcolemma at several sites, and it ends up forming what kind of looks like these half triads. Now, the sarcoplasmic reticulum does store intracellular calcium, but most of the calcium that's used for contraction, it comes from extracellular origins. Now, the sarcolemma, it also contains these pouch-like infoldings called the caveolae. And these caveolae, they contain these numerous calcium channels that opens up to allow this rapid influx of extracellular calcium. So this illustration is showing you this relaxed smooth muscle fiber. These are the gap junctions you see over here, here. These are the intermediate filaments that you see throughout this muscle fiber. These are the caveolae, these little infolds that you see here. And then you have this centrally located nucleus. In addition to that, you have these dense bodies that are located throughout in between these the intermediate filaments. Smooth muscle, it also differs from skeletal muscles in the following ways. The ratio of thick to thin filament is 1 to 13, whereas in skeletal muscle it was 1 to 2. So think about it. That's significant. It's huge. In skeletal muscle, for every one thick filament you had 2 thin filaments. In smooth muscle, for every one thick filament you have 13 thin filament. However, with that said, the thick filaments, they have heads that run along the entire length of the actin filament. And this is what makes smooth muscle as powerful as the skeletal muscle. In addition to that, there's no troponin complex that we have in smooth muscle. It does have tropomyosin, but no troponin. What does the job instead is this calmodulin that binds the calcium to the tropomyosin. Bundles of contractile proteins, they crisscross within the smooth muscle cells. So they spiral down along the long axis of the cell, which kind of looks like the stripes on a barber pole. Now, because of this diagonal arrangement, the smooth muscle cells, they contract in this twisting way. So they look like a tiny corkscrew. Smooth muscle fibers contain this lattice-like arrangement of non-contractile intermediate filaments that resist tension. They attach at regular intervals to cytoplasmic structures that are called dense bodies. Now, these dense bodies, which are also tethered to the sarcolemma, they act as anchoring points for the thin filaments, and therefore they correspond to the Z-discs of the skeletal muscles. During contraction, areas of the sarcolemma between the dense bodies, they bulge outward, giving the cell this puffy appearance. So in this illustration, you can see over here, areas of the sarcolemma between these dense bodies, how they bulge outward. Now in this illustration here, again, so this is showing you the type of muscle tissues that we talked about. So remember, we talked about skeletal muscle, and we said that skeletal muscle is found attached to bones, and cardiac muscle we only find in the only organ we find it is in the heart. It makes up the walls of the heart. And then we're talking about smooth muscle now. That smooth muscles we find lining hollow organs. And the cell shape, so you have single, very long, cylindrical, multinucleated cells with obvious striations in skeletal muscle. In cardiac muscle, we also have this striated appearance, but you have either 1 or 2 nuclei, so they're either uni or binucleate. And again, these are branching chains of cells that we have in cardiac muscle. In smooth muscle, you have single cells, fusiform, uninucleate, and they lack striations. So here we go, over here. So this chart over here, it's a comparison of your skeletal, cardiac, and smooth muscle. So this first area that we're going to be discussing now is connective tissue components among the three. So in skeletal muscle, it has epimysium, perimysium, and endomysium that's present. Now in cardiac muscle, the endomysium is attached to the fibrous skeleton of the heart. And in the smooth muscle, we only see an endomysium. Okay. And the presence of myofibrils composed of sarcomeres, as we've learned already, yes, it is present in skeletal muscles. In cardiac muscle, it's present, but the myofibrils, they're of irregular thickness. Now, in smooth muscle, there aren't any present, but actin and myosin filaments, they're present throughout, and the dense bodies, they anchor the actin filaments. Now we're going to be looking at the presence of T-tubules and the site of invagination. So we just talked right now, we just discussed that there's no T-tubules that are present in smooth muscles, and what do we have? Only these caveolae. We know that we have these T-tubules that are present in skeletal muscle. There's 2 per sarcomere at the AI junction. Now in cardiac muscle, you have T-tubules that are present. You have 1 per sarcomere at the Z-disc. Also, they're of larger diameter than those of the skeletal muscle. This further goes, and now let's see, we're going to be talking about, let's see, okay, do they have, so again, the 3 different types of muscles. Is there an elaborate sarcoplasmic reticulum? Yes, we've seen this in skeletal muscle. There is an elaborate SR. In smooth muscle, it's equivalent to that of cardiac muscle. There's some SR that does make contact with the sarcolemma. In cardiac muscle, again, less than skeletal muscle, 1 to 8% of the cell's volume. and they have these scant terminal cisterns that we see. Presence of gap junctions, absolutely not in skeletal muscles, and you will have them and in the cardiac muscle. And we find them at these intercalated discs. We do also see gap junctions in smooth muscle, and we will find them in the unitary muscles. Cells exhibit individual neuromuscular junctions. Yes, in skeletal muscles, we do see this. In heart muscles, no, we do not see this. In smooth muscles, they're not in the unitary muscles, but we do find them in multi-unit muscles. Regulation of contraction. We know that in skeletal muscles, it's controlled by the voluntary axon terminals of your somatic nervous system. And in smooth muscle, it's involuntary by the autonomic nervous system, in addition to hormones, local chemicals, and stretch receptors. And in cardiac tissue, heart muscle, it's also involuntary. It has this intrinsic system that regulates it. Also, the autonomic nervous system is able to control it. Hormones, it also affects it, as well as stretch receptors. Now we're looking at, let's see, source of calcium for the calcium pulse. So in skeletal muscle, it's the sarcoplasmic reticulum. In cardiac, it's sarcoplasmic reticulum, and from extracellular fluid. And the same for smooth muscle. The site of calcium regulation is the troponin on actin that's containing the thin filaments. Okay, and you have the same thing over here in the cardiac muscle. Troponin on actin containing the thin filaments, and then in smooth muscle, as we just learned, it's calmodulin, okay, in the cytosol. This is what's going to activate it. The presence of pacemaker, absolutely not in the skeletal muscle. Yes, we have this in the heart muscle, and in smooth muscle, it's present if it's a unitary muscle. And remember, unitary muscle is also where you only see this intercalated disc. Affected nervous system stimulation will lead to excitation. In cardiac, it could lead to either excitation or inhibition, as well as in smooth muscle. You end up getting both excitation or inhibition. The speed of contraction, it could be slow to fast. In cardiac muscle, it's slow, and in smooth muscle, very slow. Is there rhythmic contraction in skeletal muscles? No. In the cardiac muscle, yes, thankfully it is. In smooth muscle, there is rhythmic contraction in unitary smooth muscle, if the smooth muscle is unitary muscle. Response to stretch, so contractile strength increases with the degree of stretch to a point. In heart muscle, in cardiac muscle, contractile strength increases with the degree of stretch, and then in smooth muscle, there's a stress relaxation response. Now, metabolism for skeletal muscle is both aerobic and anaerobic. In cardiac muscle, it's aerobic only, and in smooth muscle, it's mostly aerobic. Now, they're able to contract in this way because of the electrical coupling of the smooth muscle cells by gap junctions. These gap junctions, they allow the smooth muscle fibers to transmit their action potential from one fiber to the next. Now, some cells, they're self-excitatory. So, for example, the smooth muscle fibers that we have in the stomach and small intestines, these are these pacemaker cells. So once they're excited, they set the contractile pace for the entire muscle sheet. Now, contraction in smooth muscle is similar to skeletal muscle contraction in the following ways. Actin and myosin, they interact by the sliding filament mechanism. The final trigger for contraction is a rise in the intracellular calcium ion level. The sliding process is energized by ATP, and the contraction stops when calcium is no longer available. Contraction in smooth muscle is different from skeletal muscle in the following ways. Now, some of the calcium still comes from the sarcoplasmic reticulum, but the majority, it comes from the extracellular space. Now, in skeletal muscle, if you remember, calcium is binded to troponin. Smooth muscle does not have troponin. Instead, it has calmodulin, and this is what the calcium binds to. Now, this activated calmodulin, it activates this myosin kinase enzyme, which we refer to as this myosin light kinase. And this activated myosin kinase, it phosphorylates the myosin heads to activate it. And this is what leads to the cross bridge formation with actin. Stopping smooth muscle contraction requires more steps than in skeletal muscles. Now, relaxation, it requires calcium detachment from calmodulin, active transport of calcium into the sarcoplasmic reticulum, as well as to the extracellular fluid. And also, it requires dephosphorylation of myosin by phosphorylase enzyme, which reduces the activity of the myosin ATPases. This illustration shows the sequence of events in the EC coupling of smooth muscles. So in this first step, what we're looking at is calcium entering the cytosol from the extracellular fluid, and we'll also have some calcium that's coming through the sarcoplasmic reticulum. Now, the calcium that it's entering through what could be voltage-gated or non-voltage gated calcium channels. Now, when the calcium enters the cell, the next thing that's going to happen is that it's going to bind itself to this protein, this calmodulin. Remember, in smooth muscle, there is no troponin. Instead, we have this protein called calmodulin. And this inactive, when this protein gets attached with the calcium, it then becomes activated. So then we call this as activated calmodulin. In the third step, we have this activated calmodulin. It goes and it activates the myosin light chain kinase enzymes. The next step, this activated kinase enzyme, it catalyzes the transfer of phosphate to myosin, and this is what's activating the myosin ATPases. In the final step, we have this activated myosin. It forms the cross bridges with the actin of the thin filament, and now we have shortening that begins. Unlike skeletal muscle, smooth muscle is like a hybrid in terms of energy usage. It uses very little energy compared to skeletal muscle. But there's always pluses and minuses to it. Smooth muscle, it takes 30 times longer to contract and relax compared to skeletal muscle. Having said that, it's able to maintain contractions for a much longer period with little energy cost, and part of that is because of the slower ATPases that we find in smooth muscle. In addition, the myofilaments in smooth muscles, they also may latch together to save energy. The smooth muscle and small arterioles and other visceral organs, they routinely maintain a moderate degree of contraction that we call the smooth muscle tone, and this happens day in and day out without any fatiguing taking place. Typically, enough ATP is made via aerobic pathways to keep up with the demand. The contraction of smooth muscle can be regulated by nerves, hormones, or local chemical changes. So let's take a look at these in detail now. In certain cases, the activation of smooth muscle by a neural stimulus is identical to that in skeletal muscle. We have an action potential that's generated by a neurotransmitter binding and is coupled to a rise in calcium ions in the cytosol. However, some types of smooth muscles respond to neural stimulation with the graded. Take 3. In certain cases, the activation of smooth muscle by a neural stimulus is identical to that in skeletal muscle. So we have an action potential that's generated by a neurotransmitter binding and is coupled to a rise in calcium ions in the cytosol. However, some types of muscles, they respond to neural stimulation with the graded potentials only. The response depends on the neurotransmitter release and the type of receptor molecules. So one neurotransmitter can have a stimulatory effect on smooth muscles in one organ, but it could have an inhibitory effect in a different organ. So for example, let's take the neurotransmitter norepinephrine. Norepinephrine will cause your blood vessels, will cause the smooth muscle of the blood vessels to contract. But the same neurotransmitter, when it's affecting the smooth muscles that's surrounding your trachea, will cause them to relax. Not all smooth muscle activation results from neural signals. Some smooth muscle layers, they have no nerve supply at all. Instead, they depolarize spontaneously or in response to a chemical stimulus that binds to G protein-like receptors. Chemical factors that cause smooth muscle contraction or relaxation without an action potential, they include certain hormones, lack of oxygen, histamines, and high carbon dioxide levels, in addition to low pH. The direct response to these chemical stimuli, it alters smooth muscle activity according to the local tissue needs, and is probably the most responsible reason for smooth muscle tone. Some smooth muscles respond to both neural and chemical stimuli. Stretching of smooth muscles also provokes contraction, which automatically moves substances along its internal tract. However, this increased tension persists only briefly. Soon, the muscle is able to adapt to its new length and then it relaxes. Now, at the same time, it still retains the ability to contract on demand. This stress relaxation response is what allows hollow organs to fill up or expand slowly. At the same time, it allows it to accommodate these greater volumes, without promoting these strong contractions that would expel its contents. And this is very important for organs when you look at your stomach or your intestines, because as the food is entering these hollow organs, it's stretching. Okay, it's allowing the food is stretching these tubes, but at the same time, these muscles are not contracting and pushing it along. It stretches and then it contracts very briefly, but then it adapts to that stretch. And because this adapts to that stretch, now your intestines, for example, it has time to further process and absorb the nutrients. If this was not the case, then think about it. As soon as food would enter, and these your stomach and your intestines, if they would just continuously contract, we would be sitting in the bathroom all the time. As soon as we eat something, within minutes, we would have to go to the bathroom. So again, this is a very important ability of smooth muscles, a very important property of smooth muscles. Smooth muscle stretches much more than skeletal muscles, and it generates more tension than skeletal muscles stretched to a comparable extent. The lack of sarcomeres and the irregular overlapping arrangement of smooth muscle filaments is what allows them to generate this considerable force even when they are substantially stretched. Smooth muscles can contract when it's anywhere from twice to half its resting length, and this capability, it allows the hollow organs to tolerate this tremendous change in volume without becoming flabby when they empty. Now let's talk about the types of smooth muscle. The smooth muscles in different body organs, it varies substantially in its fiber arrangement and organization, its innervation, and responsiveness to various stimuli. For simplicity, all smooth muscles characterized as either unitary or multi-unit. Unitary smooth muscles are commonly referred to as visceral muscles. We find them in the walls of hollow organs, with the exception being the heart. They possess all the common characteristics of smooth muscles, so we see them arranged in opposing longitudinal and circular sheets. It's innervated by the autonomic system varicosities. Often, they exhibit spontaneous action potentials, and also they're electrically coupled by gap junctions. In addition, they respond to various chemical stimuli. Multi-unit smooth muscles are located in large airways in your lungs, large arteries, erector pili muscles, and the iris of your eyes. They have very few gap junctions, and spontaneous depolarization is rare. Like skeletal muscles, multi-unit smooth muscles, they consist of muscle fibers that are structurally independent of one another. It's richly supplied with nerve endings, each of which forms a motor unit with a number of muscle fibers, and it responds to neural stimulation with graded contractions that involve recruitment. Now, it's different from skeletal muscles because, like unitary smooth muscles, it's controlled by the autonomic nervous system and hormones. All three types of muscle tissue develops from embryonic mesoderm cells called myoblasts. In forming skeletal muscle tissue, several myoblasts, they fuse to form multinuclear myotubes. And the skeletal muscle fibers, they start contracting by week 7, and this is when the embryos are roughly an inch long. Acetylcholine receptors, they spread over the entire surface of the developing myoblast. As spinal nerves invade the muscle masses, the nerve endings, they target individual myoblasts, and they release this growth factor called agrin. Agrin activates a muscle kinase, which stimulates clustering and maintenance of acetylcholine receptors at the newly forming neuromuscular junctions in each muscle fiber. Myoblasts producing cardiac and smooth muscle cells don't fuse. However, both develop gap junctions at very early embryonic stages. Cardiac muscle, it starts pumping blood just 3 weeks after fertilization. In this illustration, we can see these embryonic mesoderm cells that are called myoblasts. And what they're doing here is they're starting to divide. They're dividing mitotically. And you see these myoblasts here. What they're going to do now, now that they've divided, they're going to come together and fuse to form these multinucleate muscle fibers. Now, as time progresses, these myotubes, they mature into these mature skeletal muscle fibers. As far as regeneration of muscle goes, when we're looking at skeletal muscle, they have these myoblast-like cells called these satellite cells. And these cells, they have a limited regenerative ability to deal with dead skeletal muscle tissue. In heart cells, in cardiac muscle, cardiomyocytes, they can divide at a modest rate. But when it comes to injured muscle, we thought that, that's the end of it. Once the heart muscle is injured, it cannot be replaced. However, we are learning now that some of these cardiomyocytes, they are able to replace some dead cardiac muscle. However, it's very, very limited, and most dead cardiac muscle is going to be replaced with connective tissue. Now, again, since they just learned about this, within the past 5 years or so, they are trying to find ways to manipulate this. So eventually, perhaps in another 10 years or so, when there is dead cardiac tissue, there may be some type of therapy, uh, stem cell therapy most likely, that's going to end up repairing the damaged heart muscle. Now, smooth muscle, it has a ability to regenerate throughout life. Cardiac and both cardiac and skeletal muscles, they can lengthen and thicken in growing children. In adults, again, this is going to lead to hypertrophy. Muscular development in infants reflects its neuromuscular coordination. Development occurs from head to toe and proximal to distal. This is why a baby is able to lift his head before he's able to walk. Peak natural neural control usually occurs by mid-adolescence. Now, athletics and training, it can continue to improve neuromuscular control. Biological bases and differences in muscle mass between males and females does exist. In females, skeletal muscle makes up 36% of body mass. In males, skeletal muscle mass makes up 42% of the body mass. Now, this is primarily because of testosterone. However, men, they do have a greater ability to enlarge muscle fibers. Again, this is because of testosterone. Now, exercise, that's another thing. Because historically, men tend to do more laborsome work. Because men tend to do more laborsome work, they will have greater muscle. Whereas, so again, if you think back to hunters and gatherers, if you're out hunting, you're building, guess what? That requires a lot more muscle. So remember what we said earlier on in the lecture. You use it or you lose it. So if you're going to be using a lot of muscles, your body's going to make your muscles, and your muscles are going to be getting larger. You're going to have, again, larger muscle. If you don't use your muscle, what happens? You waste them away. The muscle mass, it gets small. So again, when you look at women that exercise, female athletes, some women will have muscle masses which are much larger than males if they are athletic or depending on the type of work they're doing. So if you look at a woman, for example, who's doing construction work, and then you're looking at somebody that's working in an office behind the desk, an accountant who doesn't go to the gym, who's not exercising, guess who's going to have, most likely, probably going to have a larger muscle mass? The female that's doing that laborious work will. Now, however, with that said, the body strength per unit of muscle mass is the same in both men and women. So what that means is the muscles that you'll find in men and the muscle that you find in women, one is not stronger than the other. They're both equal. As far as age goes with muscle, as you age, connective tissue increases, muscle fibers decreases. By age 30, loss of muscle mass starts, and this is referred to as sarcopenia. Regular exercise, it reverses sarcopenia. So again, if you're going out, you're running, if you're swimming, or if you're lifting weights, you're playing sports, this is going to help retard and delay sarcopenia. Atherosclerosis may block distal arteries, which leads to intermittent claudication. So you start seeing limping and severe pain in leg muscles. Muscular dystrophy is a group of inherited muscle-destroying diseases. Generally, it appears in childhood. Muscles, they enlarge as a result of fat and connective tissue deposits, but then they atrophy and they degenerate. The most common and severe type is Duchenne's muscular dystrophy, or DMD. It's caused by a defective gene for dystrophin. It's inherited. It's a sex-linked genetic disorder. It's carried by females and is displayed or is expressed in males. So 1 in every 3,600 births will result with a male with muscular dystrophy. Dystrophin is a cytoplasmic protein that links the cytoskeleton to the extracellular matrix, stabilizing the sarcolemma. In patients with DMD, the fragile sarcolemma, it tears during contraction, causing the entry of excess calcium. Now, this leads to damaged contractile fibers. So what you have next is the inflammatory cells that start to accumulate. So now you're going to see your macrophages and lymphocytes. They come into the surrounding connective tissue, and again, they start to aggregate there. As the regenerative capacity of the muscle is lost, the damaged cells, they undergo apoptosis, and then your muscle mass starts to decline. What you start to see in the victims is that they're becoming clumsy and they start falling more frequently. This usually appears between the ages of 2 and 7. Currently, there's no known cure for DMD. Prednisone, it can help improve muscle strength and function. They've tried myoblast transfer therapy in the past, but it didn't work out too well in humans. It did work out somewhat good for in mice model, but unfortunately not so well in humans. What they're trying now, which is looking somewhat promising, is coaxing dystrophic muscles to produce more utrophin. Utrophin is similar to dystrophin, this has been successful in mice model, as utrophin has been shown to compensate for dystrophin deficiency in mice. Viral gene therapy and fusion of stem cells with correct dystrophin genes is also showing some promise. Again, within the next 10 years or so, perhaps sooner, there might be some more. We'll be able to see what's going on with that and where they are with that. Unfortunately, patients, they usually die of respiratory failure, usually in their early 20s. And that is it for the entire series on muscles. So hopefully you found this helpful. If you like the videos, please give it a thumbs up. Please be sure to subscribe if you haven't. Also, please share with your friends. Let them know about this material that's out there. Also, if you haven't seen the first two parts, be sure to look for them in the playlist. Also, you'll find them in the links below. And if you have any questions, please email me or send me a message through YouTube, or you can leave it in the comments below. Thank you again for watching, and best of luck.